Welcome to our video walkthrough for the Carbon Prep RNA Isolation Kit. Let's walk through the protocol. First, transfer the amount of beads indicated in the protocol to a magnetic stand and allow the beads to separate from the storage buffer. Remove the storage buffer when separated, which is typically 30 seconds. If the liquid is not entirely clear, don't worry about it. Next, we need to lyse the sample. It's easiest to add the calcium and lyse the sample at the same time, so we're going to add 3.5 microliters of RPS, which is a calcium chloride solution, to 100 microliters of lysis buffer. Then we'll add the mixture of lysis buffer and calcium chloride to the sample and mix vigorously. Pipette up and down until the mixture is homogeneous. The graphene separation method is compatible with a range of lysis buffers. Both lysis buffers supplied by Life Magnetics, as well as third-party lysis buffers, can be used with the technology. After lysing the sample, transfer it to the vial containing the beads and mix. Allow the sample to sit for 4 to 8 minutes. This gives the calcium ions time to intercalate with the RNA. Sometimes you can skip, but it depends on the sample. Then, add an equal amount of ethanol. The texture of the beads will change, indicating that RNA is bound. Some protocols might ask you to use an 80% ethanol solution called binding buffer. The ethanol opens up secondary structure and is necessary for total RNA isolation. Separate the beads and aspirate off the lysate. Each RNA strand binds multiple beads, which makes the separations much faster now. You'll notice the separation only takes a few seconds. Add the Rps Wash Buffer as indicated in the protocol. Rps Wash Buffer has chelating agents that remove the calcium chloride ions. You'll perform this wash step 2 more times, but this time with regular wash buffer that doesn't have chelating agents. Between the second and third wash steps, you can transfer to a fresh vial to help minimize contamination, but this is not required. Now to get the RNA back off the graphene, we're going to allow the ethanol to evaporate and add RNA's free water. Uncap the sample and place it in a hood for 10 minutes. This drying time can depend a bit on temperature and airflow in your room. It's generally better to err on the side of a longer dry time, especially if your lysis buffer had phenol. If you end up with low 260/280 ratios, often that can be improved by increasing the dry time. Add 50 microliters of water and mix. Then place the sample in a 65°C water bath to encourage RNA release. Mix the sample by flicking it once every minute or so. RNA can also be released by mechanically mixing the sample, but mixing must be very vigorous for 3 to 5 minutes, and this method is only recommended if a robot or automated instrument is performing the extraction. For a human, a bit of heat and gentle mixing once or twice is typically easiest. After release, separate out the magnetic particles by centrifuge or by using the magnetic stand. If using a centrifuge, run the centrifuge at 10,000 RPM for 1 minute. If using a magnetic stand, keep it on the stand for 5 to 8 minutes, or until the beads are separated and collect the final elute. Third-party lysis buffers will typically require calcium chloride to be added like we did here. Some lysis buffers that contain a lot of chelating agents may not be compatible. For instance, Trizol is compatible, but Trizol LS is not. Lysis buffers provided by Life Magnetics may already contain calcium chloride, in which case you can skip adding RPS. Check your instructions. If your RNA is free in solution and lysis is not required, you will just need to add calcium chloride to the sample and no lysis buffer is required. "40 to 50% ethanol is our target for total RNA isolation, as this fully opens up the secondary structure of RNA. When adding ethanol, mix immediately to avoid high local concentrations of ethanol. In some cases, you may not need to add ethanol at all, like if the RNA is very short and has no secondary structure. This can be leveraged for size and structure separations in some cases, but see the website for details.